first thing you have to do is you have to stratify the seeds. So we take the seeds and we put them in little petri dishes so we can keep track of them, and we put them in a moist, cold climate for at least 2 months until the radicals start to push. This is simulating the apple falling to the ground and rotting before spring. If you don't do that, they don't break dormancy and they won't grow. So once we've done that, then we go ahead and plant them. This planting system was designed for us specifically for using marker-assisted selection. So they're rows of 8, just like the DNA plates are, so that we can easily select which ones we're going to keep once we've taken those samples. And I'll show you a DNA plate and how they line up after we get this one done. We then fill the tubes with soil. Now we use a pre-done mix. We use the Sunshine Mix #1. We found that it works really well. We don't have to add any additional fertilizers or anything like that. It holds moisture. It's worked out really good for us for starting the seedlings. So each year we buy brand new soil so that it's all sterile and we don't have as many problems with diseases or infestations of other insects and stuff. So that's the soil we use. We just fill these containers, just put 1 seed in each little tube. Ideally, once every seed is in the position you wanted it finally in, is like a ½ inch below the top of the tube. If it's too deep, it doesn't have enough energy to bust through the top. If it's too shallow, it doesn't have enough pressure to orient properly, and so you get the roots popping up on the top instead of. So ideally, we try to get the seed about a ½ inch below the surface. Once each tube has a little seed in it, I'll push the seeds down to that ½ inch mark so they're not right on top. And the soil is a little bit dry, so it's bouncy. So when you water in, it sinks a little bit. So I'll actually go ahead and fill it all the way to the very top, because once I water it, it compacts down. Hopefully no big chunks on top. It makes it too hard for the seed to grow through. If I see a big chunk, I try to take it off. And of course, labeling is very important. Every tray, this is a tray of 1 specific cross, so it's all this 1 cross, but labeling is key to keeping track of everything. So then we take these into the greenhouse. Water them in, see how it compacts down so they're not right on the surface. So obviously we've been planting for a while, and this is the plate that we use to collect our DNA samples. So when it comes time, these have rows of 8. There's 12 rows of 8. So we take this plate and each of these rows corresponds to a row in the plate. This way it's really easy to keep track of exactly which numbered DNA sample corresponds with the actual tube.